Good morning, everyone, and welcome to today's MPG Primer. We are excited to have Debra Van Egeren here to present to us today on the genetic histories of neoplastic mutations. Dr. Van Egeren received her bachelor's degree in computer science and molecular biology from MIT in 2016 and subsequently completed her systems biology PhD at Harvard. During her time as an undergraduate, she worked with Professor Daryl Irvine to help develop a B cell vaccine platform for treating melanoma. In her more recent experience, she's been using mathematical modeling to study heterogeneity in tumor environment and progression. For that, she was advised by Franziska Mikower. She's now starting a postdoc in Dan Landau's lab at Weill Cornell Medical School next week, and so is in process of moving. So we are especially thankful for her today for making time to share with us her exciting work. She's happy to take questions from the audience during her presentation. So please go ahead, type in the Q&A or raise your hand, and she'll answer your questions as they come. Thank you very much.
All right, thank you for that introduction and for the opportunity to present here. I've been to a lot of Broad talks over my years here, so I'm excited to be able to present one. So yes, I will be talking about neoplastic mutations in general and a couple experimental and computational techniques to analyze their histories. And I'm going to do that in the context of our kind of recent work with Sahand Hormoz, who is a faculty member at HMS and DFCI, which characterize a history of a particular mutation in this blood cancer called myeloproliferative neoplasm. So I'm going to quickly just go over a little bit about MPNs first, and then we can get into some of the more kind of more analysis type part of it. So MPNs are this group of 3 related chronic blood disorders. They are essential thrombocythemia, which is marked by like an increase in platelet counts. Polycythemia vera, or PV, which is marked by a increase in red blood cell counts, and primary myelofibrosis, which is mostly characterized by fibrosis in the bone marrow and can be kind of associated with different changes in blood counts, as well as extramedullary hematopoiesis. And in general, these 3 conditions are pretty indolent. So they have you can live for decades after diagnosis with minimal kind of symptoms and side effects from treatment. However, there is some risk for transformation to more severe malignancies such as AML, which increases as you kind of get to the myelofibrosis end of the spectrum. And it might not be super obvious what kind of ties all of these disorders together. It sounds like their phenotype is somewhat different. But one thing that does tie them together is a group of shared mutations. So the majority of cases of MPNs are caused by a single driver mutation, especially in this particular gain of function driver in JAK2, which is a kinase downstream of receptors like the erythropoietin receptor and the thrombopoietin receptor, which are kind of 2 important growth factors related to myeloid cell differentiation in hematopoiesis. So the JAK2 V617F mutation causes constitutive activation of downstream pathways from these receptors and causes the vast majority of PV cases, as well as many cases of the other subtypes. You also have other mutations that are associated with disease, most notably a mutation in the calreticulin receptor, the calreticulin. And you can also have secondary mutations that are also found in addition to these kind of primary MPN causing mutations, many of which are associated with other blood malignancies like AML. Most notably, we can see a fairly reasonable fraction of patients showing secondary mutations. Often they're subclonal in a TET2, for instance. And so one thing that we wanted to kind of investigate was how this JAK2 V617F mutation was causing disease in patients. So this mutation has been studied to a fair extent in mouse models, for instance, and in like in vitro experiments or in the transplant setting. But it's been a little bit more challenging to study this within its native context within MPN patient bone marrow. And to kind of look at this, we did two related kind of sets of experiments and analyses. The first one I'm going to talk about is doing joint single cell genotyping and transcriptomic profiling of bone marrow from these patients to figure out how this mutation directly affects cell phenotype. And the second is that I'm going to go over is looking at kind of more the history of expansion of the hematopoietic stem cells carrying this mutation using single cell whole genome sequencing and phylogenetics. So in the past, it's been a bit challenging to study the direct effect of this mutation on cell phenotype because there are no good like surface markers to say sort between cells with the mutation and without the mutation within the bone marrow. So you can't directly compare the two within a patient. To help us kind of study this mutation, then we developed a protocol, which is similar to some other protocols developed, say, by the Landau lab, to jointly profile the transcriptome and genotype individual mutation loci within individual cells in a population. So to do this, what we did was we took bone marrow biopsies from 7 newly diagnosed MPN patients, and we isolated the CD34 positive hematopoietic stem and progenitor cells, some of which carry this mutation. And then we kind of started out by creating a normal sort of 10x library, doing the first few kind of cell barcoding steps within that protocol. So you ended up with the cDNA library that contains the whole transcriptome of all cells, including the JAK2 gene. And some of these JAK2 transcripts obviously will have mutation in them. And so what we did next was we split this library into 2 pools and did specific amplification of the JAK2 mutation locus using a set of primers we designed on one part of this library. And on the second part of the library, we just did normal single cell RNA-seq. And so by matching these cell barcodes, you can figure out which cells have the mutation, or at least are expressing transcripts that carry the mutation. And then you can match that to the whole transcriptome sequencing data using those barcode matching. So in general, this type of protocol is a little bit easier if the mutation is closer to the 3' end of the transcript. In this case, unfortunately, we're around, I think it was like something like 1,500 base pairs from 3' end of the JAK2 gene. So it was a little bit more difficult to kind of amplify out this mutation specifically. And obviously it requires that the gene that has the mutation is expressed in the cell population of interest. So it turns out there is some differences in JAK2 expression between different cell types in our population, which I'll show in a minute, which does limit our ability to assess the frequency, say, of the JAK2 mutation in specific cell types in the bone marrow. So we can take a look at the 7 patients that we sequenced. We sequenced 3 with ET, which is the subtype that has platelet overproduction, or 3 with ET that have the V617F mutation, 1 patient with ET that had this non-canonical mutation in the same amino acid, but in the same amino acid as the canonical V617F mutation, but it's a different uncharacterized mutation. So that was kind of an interesting has some interesting biology that we're going to go through in a minute. And then 3 patients with PV. So of note, 2 of these patients have significant populations of cells with a TET2 mutation. which we did also sequence, but unfortunately our capture efficiency was a little bit lower, so we don't have a full analysis on that. But we're able to sequence around 6,000 to 8,000 CD34 positive cells per patient. And we were able to capture at least one JAK2 transcript in around 5 to 15% of, or in around 10% of cells in each patient. And so we can take a look just at the transcriptome data at first, and then we'll overlay on that mutation data in a minute. But we can see that with the CD34 positive enrichment, we have kind of all lineages of the kind of the hematopoietic differentiation hierarchy here. We have the top HSCs going down into the erythroid and megakaryocyte branches, which are the ones that are kind of expanded in this disease. We have granulocyte and macrophage progenitors here, as well as lymphoid progenitors. So if we take a look at this example patient, we can see that the cells that carry a JAK2 V617F transcript are kind of on the left side of these plots. So they're the side associated with the erythroid and megakaryocyte progenitors, whereas cells that carry just the JAK2 wild type transcript are more evenly distributed throughout the plot, though, as I said, there are some differences in the overall expression of JAK2 in these cells. But this is consistent with our understanding of the disease phenotype, that kind of this JAK2 V617F mutation is likely causing expansion of progenitors along this erythroid megakaryocyte trajectory, which leads to this overrepresentation of cells with the mutation over in those lineages. We can take a look at all of the data from our patients together. The top line, again, is colored by cell type, whereas these bottom two lines are the cells with the JAK2 wild type transcripts and mutant transcripts, respectively. And you can kind of see, if you scan over it, that most of the red dots here are kind of to the left side of the plots. So they, the cells with mutant transcripts are associated more with erythroid megakaryocyte lineages than you'd expect from just the kind of overall expression of JAK2. One thing to note that's sort of interesting here is that there's no super clear differences. This is a little difficult to tell, but there's no super clear differences between patients with the two different disease phenotypes. So it's not obvious that a patient has ET or PV, even though they have different clinical symptoms. So it's still a bit of a mystery, at least from this analysis, as to how this mutation leads to these different, different disease phenotypes here. You can also see a kind of a difference in this pattern if you focus on this patient with the non-canonical V617L mutation. And you can see that the cells that carry the mutant transcript are more evenly spread throughout the hematopoietic hierarchy here. That distribution pretty well matches the distribution of cells that just express wild type JAK2 transcripts, indicating that this mutation, in contrast to the V617F mutation, doesn't seem to create a fate bias in these cells. So it doesn't seem to preferentially expand cells that are creating the red and blood cells and platelets. So it's a little bit unclear as to how disease is kind of being caused in these patients. So we were able to essentially use this technique to amplify the disease-causing mutation in this cancer and figure out, one, we could quantify how this mutation was affecting the fate bias of these kind of these HSCs with the mutation. Another thing we could do is we can directly compare the phenotypes of these cells. Particularly, we can take each cell type and we can compare the cells with the JAK2 with a JAK2 mutant transcript and compare them to cells with a JAK2 wild type transcript and do differential expression essentially, and figure out what the direct effects of the mutation are on the transcriptome. And the pattern that we kind of saw across the board, I'm showing the results for our ET patients combined, is that we see increased expression of HLA genes or antigen presentation genes in general in these cells, which are likely a part of a larger pattern that is associated with upregulation of inflammatory genes or interferon response genes, of which these antigen presentation genes are just some of the most highly expressed. We also, interestingly, see increased expression of the lectin receptor in these mutant MAPs, or megakaryocyte erythroid progenitors here. And that's interesting because the leptin receptor has been found to also regulate hematopoietic stem cell stemness in other contexts. So we're kind of following up on that finding as well to see if that's important in the pathogenesis of MPNs.
Deborah, that's a really interesting observation with what's known about leptin and kind of appetite and metabolic regulation. Does this tie in with any kind of patient phenotypes or risk factors?
Yeah, so it's a little bit unclear how much of the leptin receptor function in bone marrow crosses over with the leptin receptor function in like regulating, as you said, appetite and stuff like that. Because leptin itself is kind of synthesized in the bone marrow as well by like stromal cells and things like that. So I'm not quite sure if it's been worked out how those two kind of sources of leptin and functions of leptin crossover. So I guess I don't have a satisfying answer for you, but yeah, unclear. All right. And the kind of last thing I think I will say about the our differential expression results here is to note that we saw kind of this curious population. If you noticed on one of the previous slides, we saw this kind of curious population of cells that are expressing CD14, which is a marker for monocytes. And it turns out they're not expressing CD34, which is interesting because they passed our CD34 positive selection protocol. So we think they might be just maybe as like a sticky cell that kind of ended up carrying through the experimental part of this. But anyway, in any case, we found this population of CD14 positive cells that we think are bone marrow monocytes. And we found that they actually had a fairly high fraction of cells that were JAK2 mutant. And if you kind of compare the JAK2 mutant wild type cells within the CD14 positive monocyte compartment, you see a lot of differentially expressed genes. Again, they are related to antigen presentation, the interferon and inflammatory responses, and it turns out they're also associated with this intermediate monocyte phenotype, which is associated with antigen presentation as well as expression of certain cytokines. So it's interesting to think that these, since we know that in MPN cytokines are somewhat important in terms of pathogenesis, it would be interesting to kind of follow up on this and figure out whether these mutant monocytes specifically are driving kind of any sort of disease phenotype in these patients. And in particular, we also saw that you see increased expression of this cell surface receptor SLAMF7 in JAK2 mutant CD14 positive cells in these patients. And this is particularly interesting because this cell surface receptor has been shown in myelofibrosis to be functionally associated with the fibrotic response in these patients. So they're directly depositing things like collagen to create bone marrow fibrosis. So we're also following up on this observation here to figure out whether in these patients who don't have overt fibrosis, whether these SLAMF positive monocytes might be a marker of kind of marker for risk of progression to fibrosis, or B, some sort of cell population that can be targeted, say with the monoclonal antibody against this receptor to reduce the chance of progression in these patients. So that is kind of wraps up the first part of the study that we did, looking at kind of studying the direct transcriptomic effects of these of this JAK2 V617F mutation on cells in the MPN bone marrow. I'm going to stop here if there are any questions in this first part, because I will be kind of pretty abruptly changing gears to the second part of the analysis.
There is a question that says, cool work. Have you thought about if ambient RNA, particularly ambient RNA and JAK2, might be an issue in this system?
Yes. So ambient RNA is possibly an issue here, though it's unclear. We didn't really directly test for this. I mean, it could be an issue. I mean, the quality of the single cell RNA-seq in general was pretty good in this data set. So there weren't clear markers of like RNA contamination within the 10x runs. So that's one thing. We did do some controls on this. So you can just take a kind of cell line with and without because the JAK2 genotyping obviously is just done to identify cells with and without the mutation. So we did do some cell line controls to figure out how well we can discriminate against those two populations. So there's that. But to be honest, we didn't kind of directly control for RNA contamination in any more specific fashion.
Great, thank you. And then I was wondering, many times we capture a point in time with a disease state that might not be kind of your end disease state. And how do you kind of think of that and contextualize that in your thoughts about your results?
Yeah, there's a couple things about that. Right, so all of our patients here were this part of the analysis was done on patients who are newly diagnosed. So that's one particular, as you were saying, one particular point in time. I'm going to show you in this next part things about we did an analysis focused on figuring out what happened before diagnosis, but not obviously, but it was more difficult to figure out what the kind of phenotypic effects are. We could figure out things like history of expansion and things like that, but in terms of transcriptomic phenotype, it's a lot more difficult to figure that out. In theory, right, you could do repeated biopsies on these patients to figure out how their the phenotype of the transcriptional phenotype, for example, changes after diagnosis, right, and after treatment. So most of the treatments for this disease are not disease modifying. So in theory, right, that would, a lot of the serial kind of sampling would mostly capture disease-related changes associated with progression, which could be pretty interesting. And you could also capture additional changes if the patients acquired secondary mutations during the course of the disease. So that could also be an interesting thing as well to study. So yes, the answer to the question, I suppose, is that, yeah, you see only this kind of point at diagnosis, but you could sample later and figure out how that changed, for instance.
Thank you.
All right. And so that was an excellent segue into the second part here of the analysis, looking at, as I mentioned, kind of the history of disease before diagnosis in these patients. So one kind of source of data about the history of cells is kind of their genetic history, right? So mutations, so like somatic mutations can essentially permanently encode information about the expansion history and sometimes other features of a cell population over time. And so what we did was we took we again took bone marrow biopsies from 2 of the patients in our cohort, and we performed single cell whole genome sequencing. To be more precise, we performed single colony whole genome sequencing on these hematopoietic cells in order to get more genetic material out of each hematopoietic stem cell that we were sequencing, so we could get better sequencing results out of them. So what we did was we took bone marrow biopsies from 2 patients. We sorted out hematopoietic stem cells from these patients, and we cultured them in kind of this matrix ex vivo for, I think it was like a few weeks, so it was like 4 to 6 weeks. And we expanded them into colonies derived from each individual cell. And then we sequenced each colony. And so each colony essentially represents an individual hematopoietic stem cell from each patient, and we were able to use those data to reconstruct lineage trees from each sample from each patient. And as you'll see, there are kind of 2 parts of each lineage tree that have kind of pretty distinct shapes, and so we can distinguish between the JAK2 wild type cells in each tree, as well as the cells with the JAK2 mutation, so this cancer clade. And we're going to use the shapes of each of these, subtrees and use those to figure out something about the history of expansion dynamics in HSCs with and without the mutation. But first, we can just look at by eye the shapes of these trees that we're getting. So we're sequencing around, say, around like tens of cells from each patient. So again, we sequenced 2 patients with ET from our previous data set, one who's 34 and one who's 63. And we can see a few immediate things if we just inspect these. First, you can see that the cells with the JAK2 V617F mutation all sit together on the tree, and they sit together on this subtree that has a more recent common ancestor than the common ancestor of the entire population of cells that we sequenced. And we can also see that the overall number of somatic mutations in each of these cell lineages, which represents the number of somatic mutations from kind of away from the zygote that each individual cell has. We can see that this, if you look at the y-axis scales here, that in this younger patient, you have around 700, say, mutations in each of these individual cells that they've accrued over their lifetime, whereas you see a higher number of mutations in this older patient. And this is consistent with the hypothesis that essentially cells gradually accumulate mutations over time, most of which don't have much of a function, but we can use these mutations sort of as a clock to be able to time events in the history of these cells. We can also take a look at the mutational profiles of each of the cells and of the entire population as a whole. And we can see that we have a fair number of mutations that are associated with these signatures, especially 1 and 5, that were defined by sequencing a bunch of these are the kind of these somatic or these mutational signatures that have been previously defined by other groups. We can see that 1 and 5, for instance, which are associated with sort of clock-like processes, are sort of enriched in these data as well. And there's no additional signatures that are associated with specific carcinogens or anything like that. But anyway, so we're going to use these mutations to start out as a timer for a particular molecular event. In particular, we're going to figure out at what time this most recent common ancestor of all of the JAK2 mutant cells existed. And by using this kind of branch length here, we can infer that the driver mutation for these 2 patients occurred decades before their diagnosis. So in this case, this mutation occurred, what was this, 25 years before their diagnosis. And in this patient, I think there's even a longer period between the JAK2, or the original JAK2 mutation and the diagnosis of disease. So that's an interesting finding that was a little bit surprising. It suggests that essentially you have this pretty long phase in which this mutant population essentially lays not necessarily dormant, but undetected in these patients and doesn't cause symptoms. And this is consistent with the idea that some people have clonal hematopoiesis that has this JAK2 mutation in it. So clonal hematopoiesis is this phenomenon that where individuals, particularly older individuals, have detectable levels of leukemia or malignancy associated mutations in their peripheral blood, but don't have overt disease. They have a higher risk of getting certain blood cancers, but they aren't currently showing symptoms. So it's possible that, say, for years, these patients can have no symptoms, have a JAK2 mutation, and have essentially clonal hematopoiesis. And this raises a question as to whether maybe there could be an early detection or an early detection protocol for identifying these patients who are at risk for transformative 2 disease by looking at their kind of peripheral blood mutation status. This finding has been followed up and replicated by this other group. This is Jyoti Nangalia's work. That's a preprint currently, and they sequenced kind of a lot more patients, but also they sequenced patients that had additional, a more complicated genomic profile. So they had additional secondary mutations, but they also found that in general, the JAK2 mutation occurred pretty early in the lifetime of these patients and several decades before diagnosis. In this case, for example, the mutation occurred within around 1 year of the patient's birth. So this disease has this long preclinical phase. The other thing we did here is we used the shape of the lineage trees to more precisely pinpoint and infer how the history of expansion of HSCs with the mutation in these patients. And we did this by employing coalescent theory, which is this technique from evolutionary biology and population genetics to infer population sizes through history using phylogenetic trees. And I will give you the kind of hand wavy general form of this idea. And then if you're interested, you can go back and follow up on our paper, or I'll cite this other paper that does this in normal hematopoiesis, which is a good read as well. But in general, you can think of this intuitively as follows. So if you imagine if you had a really small population on, let's say you had like 20 people on an island, and you sampled 5 people from that small population here, they would be, and this is like an isolated island with very little immigration and emigration from it. Those 5 people would be likely pretty closely related and would have a common ancestor, which is represented by kind of this node at the top of the tree, where all of these 5 individuals kind of coalesce here. They would have a common ancestor that's relatively recent. But if you took the entire population of the US, say, and you picked 5 random individuals from it, you would find that they are fairly genetically distinct, more genetically distinct from the people on the island. And they would have a common ancestor that traces back quite a bit farther in time. So they would have this kind of longer type tree if you reconstruct their phylogenies versus this smaller population here. So in general, this is just to show that the shape of a phylogenetic tree encodes something about the size of a population over time. And you can extend this principle to look at how the population size varies over time, essentially. So we're going to use, and you can do this kind of, this is a very, again, hand-wavy explanation of it, but there are kind of more precise algorithms that do this quantitatively. One of those algorithms was used in this paper from Peter Campbell's group in 2018, I think, to characterize wild type HSCs just from like a normal individual. And they were able to essentially infer the size of the HSC population over time in this individual. And they found, essentially estimated that you ended up with like a pool of around 100,000 HSCs with a person, for instance. So they essentially did this with this kind of wild type part of our data. And what we're going to do is we're going to use the same algorithm to infer the history of population expansion in this in the JAK2 mutant cells here, which you can already see have a distinct population kind of, or a distinct phylogenetic structure from these wild type cells here. We did this for our 2 patients, again, the younger and the older patients from the trees that I showed you before. And we can see that, for one, that the number of mutated HSCs grows over time in these patients. You can also see that, in particular in this patient, for example, that the number of mutated HSCs that we infer stays reasonably low. We're talking about like less than 1,000 in this patient over the first decade of their history with the mutation. So there's this kind of like, again, this long period where you have relatively few cells with the mutation not causing disease, and they're just kind of hanging out in the bone marrow. But they eventually expand out and then do cause overt symptoms. So yeah, so we see similar patterns in kind of both patients here. And finally, what we did with the data to infer kind of the rate of expansion of these HSCs over time in a different way is we used population simulations, and we these Wright-Fisher population genealogy simulations, and we used ABC, approximate Bayesian computing, to figure out what expansion rate or fitness advantage for the HSCs with the JAK2 V617F mutation best match up with the lineage data that we've collected. So the way we did this more precisely is we used these Wright-Fisher simulations to simulate the history of population expansion in these cells. We also simulated the mutational process using the mutation rates that we kind of inferred from our lineage trees. And then which essentially allows us to simulate phylogenetic trees for different fitness values for the JAK2 mutated population. We can then compare the simulated phylogenetic trees with the real phylogenetic trees by collapsing them onto this thing called lineage through time graph. The particulars aren't super important, but you can compare trees with each other, and then we essentially then inferred the fitness advantage of the JAK2 mutant HSCs in our data set. And so again, for our 2 patients, the most important thing here is we were able to construct a joint distribution for the most likely fitness advantage for the JAK2 mutated cells here. So the colors here on these plots are the joint distribution probability here. And we found that in this patient, you ended up with a fitness advantage of around around 63%. So essentially cells were dividing 63% more than the wild type HSCs. And for our other patient here, we found a slightly lower fitness advantage. It was around 45%. So we were able to essentially use these simulations to infer a fitness advantage for these HSCs in vivo without in the kind of context in which this mutation it plays a role in disease. And finally, I just wanted to close out this section by mentioning that this principle of using phylogenetic trees to infer things about cell dynamics is not limited to just learning things about expansion dynamics, for instance. This paper from Dan Lando's group that came out recently in Nature Genetics, for instance, used lineage trees inferred from single cell methylation data to infer the rate of cell state switching in GBMs. And essentially, kind of the principle you can go by is you can look at kind of the lineage histories of a population of single cells, and you can figure out how closely related the lineage history is with the cell state, the current cell state. So if cells in a particular clade are all of the same state, then cell state switching happens more slowly than if, like, say, cell states are kind of mixed throughout the leaves on the tree. So you can more precisely quantify it, and they kind of more precisely quantify it in their paper, but that's another principle you can leverage for looking at the history of cell dynamics in a population using single cell sequencing. So the very last thing I will mention is this sort of non sequitur extension that we performed to kind of additionally show the value of having single cell whole genome sequencing data to go along with transcriptomic data, for instance. So if you remember, if you looked at the lineage trees that we generated, there are many mutations that mark this population of JAK2 mutant cells. So there are additional mutations in this branch here that are common to all cells with the JAK2 V617F mutation. And then there are additional mutations that are specific to individual HSC clones that we sequenced. So both of those are also associated with the JAK2 mutation that we sequenced and are specific to each patient. We can also, so we can use these mutations to identify additional cells that we might have missed from the initial analysis on the JAK2 mutation specifically, that lie within this clade. For instance, we can sequence, say, UPF. So we found this mutation in UPF1 that was on this branch here that was common to all JAK2 mutant cells in patient ET1, for example, and we're able to use a similar PCR approach to amplify that mutation in the transcriptomic data from our single cell RNA-seq part of the study. And that greatly increased the number of cells that we were able to confidently identify that are within the kind of cancer cell population. The other thing we did was we took these single clone mutations and we kind of looked for them specifically in the 10x data. It's a little bit difficult to do that because the 10x data obviously is 3' enriched, and it's pretty difficult often to look for specific somatic mutations. But we managed to do that in a limited set of mutations. And combined, we were able to essentially increase the number of cells that we're able to identify as being part of the cancer population versus the wild type population in our patient bone marrow samples. And this raises the possibility that you might be able to do single clone analyses in the transcriptomic data, for instance. So if you're able to identify enough cells that are associated with individual HSCs, say in our single cell whole genome sequencing data set, you could study the output of individual HSCs, for instance, and figure out, whether different HSCs have higher or lower differentiated cell outputs and things like that. This is just an example of what we could do with our data. We found that, say, each HSC in our data set had slightly different numbers of cells in the single cell RNA sequencing data, for instance. It's a little bit difficult. The detection rate for these individual mutations is pretty low, so it's a little difficult to actually do a good quantitative analysis here, but it's an interesting thing to think of for the future. So that kind of wraps up the talk here. I'd like to acknowledge everybody who helped out with the study, particularly Sahand Hormos in the Hormos lab. The experiments that I showed here were not done by me, they were done by Max and Sean. And Javi helped out with some of the lineage reconstruction and analysis as well. And this was a collaboration with many clinicians, for instance, at DFCI and Brigham. And as well as other labs, both at Harvard and abroad. With that, I'd like to thank everybody for being here, and I see there's at least one question here, so I'm happy to take questions.
Great, thank you so much, Deborah. There is a question, which
Yes.
was, which other somatic mutations commonly associate with the presence of the JAK2 V617F?
Yeah. With our two patients, we didn't see, it's hard to figure out if there's any additional somatic mutations that are more likely to be associated with the V617F cells, for instance, because we only have two patients. So, and none of the mutations occurred in both patients. None of the additional somatic mutations occurred in both patients, so we couldn't say if there was an enrichment of a certain mutation that's particularly associated with the cancer clone. In general, the somatic mutation we saw, we thought were essentially random ones and were benign. None of them really show, we ran them through a couple databases of known driver mutations, for instance, and none of them really came up as associated with any sort of cancer, and most of them were as not in coding regions or not predicted to have really any effect on function. So we're thinking they're just essentially random somatic mutations. To kind of complete the answer to that question, right? There are some. In general, on the population level, there are some mutations that are associated with JAK2 mutant myeloproliferative neoplasms. I kind of went over them quickly at the beginning. They're other leukemia-associated ones. And we did see some of those in the other patients in our data set.
Thank you. And there's another question as to whether lineage tracing can be applied in WES or WGS data.
Yeah, so I think either of those would work. So the lineage reconstructions we did were on whole genome sequencing data. We didn't try to subsample to whole exome because we could just sample the exome and see how that changes the trees that we get, for instance. I suspect they would be pretty similar since we were getting hundreds of mutations per lineage, which, yeah, it would go down a little bit if you did only exome sequencing. But in general, you could try it with exome sequencing, and I think you'd get a decent result, but whole genome sequencing would just give you better resolution.
And there's two more questions. One of them is to know if you have evaluated how the variant allele frequency of JAK2 influences the outcome.
Yeah. So in terms of influencing disease outcome in general, there is some association of that between VAF and disease like prognosis, but it's pretty weak. And most of the treatments for, especially for ET and PV, don't really affect the variant allele frequency of JAK2 at all, but still are reasonably effective in eliminating symptoms. So VAF is not like the greatest target clinically for marking disease progression and prognosis. In terms of our study, the one interesting thing I'll note that I kind of took out of here is that we found a somewhat weak association between peripheral blood VAF, which is mostly the VAF in myeloid lineage cells, so like granulocytes, macrophages type cells. We found a weak association between that VAF and the VAF in HSCs. So that might be one factor in kind of this weak association we see clinically in terms of prognosis, because the most common clinical measure of the variant allele fraction does not actually correspond particularly well to the actual kind of root variant allele fraction in the stem cells in the bone marrow. That's an interesting complication as well.
Thank you. And then there's one last question, which starts off with great talk. Any thoughts on if these results are likely JAK2 specific, or if you might see similar results with CALR, et cetera?
Yeah. So in terms of CALR mutations, in terms of the first part of the talk, in terms with the transcriptional phenotype comparison, there are differences. You can see, I think Dan Landau put out a paper in Nature a year or two ago looking at the CALR mutation specifically using a similar technique as we showed here at the first part of the talk, and found that, for instance, the unfolded protein response was particularly important in CALR mutant cells in MPN patients, for instance. In terms of the second part of the talk, looking at the lineage histories and the expansion histories, I would expect to see something similar, but I actually don't quite know, right? So I'm trying to think, because a lot of the in vitro and in vivo studies have focused on the JAK2 mutation in MPNs. I'm trying to think if anybody's measured side by side in vitro, say the division rate, for instance, of HSCs with the CALR mutation versus JAK2 V617F. I'm not sure anybody's done that. So I'm not sure, I don't know whether I'd expect, say, a longer latency period in CALR mutant patients rather than JAK2 V617F patients. The other thing I would say is I don't think progression dynamics are super different between CALR. It's not like CALR mutant patients like progress super quickly or anything like that. So maybe you would see a similar sort of latency period in terms of at first approximation. Yeah.
And there was just one last question, which is, have there been cardiovascular phenotypes in those two patients that you know of?
I don't think so. I don't. To be honest, the clinical data that we've gotten is mostly related to just the cancer history and and we didn't see any. There wasn't, in particular in these two patients, there wasn't any additional oncology history to speak of. So I actually don't know about cardiovascular complications in those two specific patients. I assumed the question was related to the relationship between cardiovascular complications and say a CHIP or clonal hematopoiesis that's been found in other studies. That would be an interesting thing to look at. Yeah, sorry, no good answer.
So much. This has been an excellent presentation, and best wishes for your future postdoc.
Thank you. Thank you, everybody.